My name is Jenny Guerin. I work at the South Australian Seed Conservation Centre, along with Dan Duval and Tai T. And today I'm going to talk about some of our work on orchid recovery projects since the 2020 fires. There were several fires in South Australia in late December to early January last year, and the largest was on Kangaroo Island, where nearly half of the island burnt over several weeks, mostly on the western end of the island through the Flinders Chase National Park. Caladenia ovata, also known as the Kangaroo Island spider orchid or the ovate spider orchid, was one of the target species for the Bushfire recovery project that we became involved with Nushka Reiter from Royal Botanic Gardens Victoria and Ryan Phillips at La Trobe University. The orchid has a characteristic ovate shaped labellum and upswept petals and sepals. The KI spider orchid is listed as nationally vulnerable and occurs mainly on the island, with a few records from Dean Creek Conservation Park on the mainland. This map shows the data from the SA database with several dots where the orchid has been recorded, but I've added the dates on this slide to show that a number of these records are historical and the orchid has not been seen for over 100 years in those locations for some cases. We searched the island following up old records, and also we looked in the fire scars for evidence of the orchids to gain a better understanding of the current status of this species. The searchers were greatly assisted by botanists on the island, with a special mention of Dr. Andy Young. This table shows the total number of flowering orchids that were located on the island during the spring of 2020. There were less than 100 flowering plants found in total, and they were mostly in two locations that were on the eastern end of the island, in Bay Area Conservation Park and Lashmar Conservation Park. We searched around recorded locations in the burnt areas in Pandana Conservation Park and also near the Stunsdale Boom River in the southwest, and we saw many other orchid species in flower, but we didn't find any of the Kangaroo Island spider orchid. The habitat at Bay Area and Lashmar was broom bush and thryptomene, and the orchids were mostly found sheltered from herbivores within the shrubs. Several plants that Andy had located and tagged in flower a previous day were all eaten a day later when we went and looked. Due to the high levels of herbivory, we had to cage the remaining flowers that were seen in chicken wire cages, and we hand pollinated each flower so that we could get seed development for collection. There was nearly 100% pod set after hand pollination, and the seed viability was high from both collections. In total, over 600,000 seeds were collected from these two populations and banked in the South Australian Seed Bank. Mycorrhizal fungal isolation was undertaken from Caladenia ovata leaf collars from Lashmar Conservation Park. We took these back to the lab in Adelaide, and this slide shows that the fungus was effective with healthy protocoms grown on seeded agar plates. The protocoms have since been grown on in flasks and have now been potted out into soil in the shade house that we have at the Adelaide Botanic Gardens. And these orchids will hopefully be ready for translocation in a couple of years' time. This is a photo of the site at Lashmar Conservation Park. We did some work trying to identify the pollinator for Caladenia ovata. It might be interesting because there aren't that many orchids or spider orchids that have that morphology of the upswept petals and sepals, and there might be a unique pollinator association. This is a photo of Andy, who is actually firstly an entomologist, and he spent several hours on sunny days sweeping with a net and closely observing flowers, but we have been unable to identify the pollinator as yet. Another orchid that we worked on Kangaroo Island for the T Reid Project was Thelymitra Methusii, which is commonly known as the spiral leaf sun orchid. And you can see the very distinctive spiral leaf in the picture here. This map shows the distribution of records for Thelymitra Methusii in South Australia. It's listed as nationally vulnerable, also occurring in Victoria and Western Australia. And in South Australia, it's listed as endangered at the state level. It's found on Kangaroo Island and in the southeast region. For this project, we focused on the Kangaroo Island populations. This map shows a close-up of the records on Kangaroo Island. They were all on the west end of the island, and there were 5 locations in total where the orchid has been recorded. All of these locations were searched post-fire, but it was only found at one location, and that was the one near Goss.
The fire scorched this area, and the small tiny spiral leaves were difficult to pick out in the fire scar. Here is Andy and Ty searching along the track for the leaves. This was in August where most of the plants were found, but they were already finished flowering. Further searching in September, we didn't find any more plants. So searchers went for over 10 k's over 2 days looking for the leaves. 20 plants in total were found. They were all caged until revisiting for seed collection. All the plants that were found were on the edge of the track where the intensity of the fire was a little bit less than in the surrounding scrub. One of the tubers was dug up to measure the depth of the tuber beneath the ground, and it was found to be only a couple of centimetres. So we wondered whether in the sandy soil, whether the intensity of the fire was enough to destroy the orchids, or perhaps these orchids were found on the track because they'd been there in response to disturbance. When we came back to collect the seeds, there were only 3 pods that were collected, yet this gave enough seed for some germination testing, and there's also some that have gone into long-term storage. We obtained a fungal isolate from root tissue, but it wasn't effective at germinating seeds, and this year we hope to get some fungus for seed germination, and also to look for the populations while the orchid's in flower, which will be a lot easier than looking for it at the leaf stage. The next orchid that I'll talk about is the black-beaked duck orchid, or Caleana disjuncta. We're working on this as part of a project funded by the T Reid Foundation for bushfire recovery of 4 threatened orchid species on Kangaroo Island. There were only 2 previous records on the island, and so we were interested to see what numbers of plants we could find post-fire. So it turns out that this was another great example of working with Kangaroo Island botanists, and after we sent out information that we were looking for this duck orchid, 3 new populations were found in burnt sandy mallee around the Goss district. Hundreds of plants were observed, and we hand-pollinated and caged 2 populations and have banked nearly 1 million seeds. Mycorrhizal fungi was also isolated from plant tissue, and we're now experimenting, very hopefully, that we'll be able to grow some duck orchids in the lab. At the same time as the Kangaroo Island fires, there was also fires on Eyre Peninsula in the northwest and the southeast of the state, and also in the Adelaide Hills. The Cuddly Creek fire burnt approximately 23,000 hectares of the Adelaide Hills, including towns, properties, and through conservation parks. Pterostylus cuculata subspecies silver-cola, or the leafy greenhood, has a restricted distribution in the Mount Lofty Ranges. It's listed as endangered in South Australia and nationally vulnerable. There was a population of about 100 plants in the Lobethal Bushland Park that was burnt in the Cuddly Creek fires. And since those fires, the greenhoods have not emerged in the previous year. But luckily we had some seeds banked from previous collections of that population, including one that was just before the fires in 2019. We've been monitoring some of the threats to these leafy green hoods in another population at Belair National Park. Camera trap footage has shown that the green hoods are browsed by kangaroos, and pollen exclusion experiments have showed that these orchids don't no pods were formed after bagging flowers. And weeds are also a threat to these leafy green hoods. Fortunately, we have been able to propagate some plants through using symbiotic seed germination, using seeds from the historic collections from Bushland Park. Plants that were grown from seed last year are already flowering in the shade house this year, and this season's plants are growing well. We expect to have hundreds of plants available to translocate into the Lobethal district over the next few years. So lastly, I'd like to acknowledge two of the sources of funding that we've had for this work. They are the Wildlife and Habitat Bushfire Recovery Program that's in collaboration with La Trobe University and with Ryan Phillips and Nushka Ryder from Royal Botanic Gardens Victoria, and also the John T Reid Foundation. I'd like to acknowledge most of the photos that I've used have been taken by Dan Duval or Denzel Murfitt, and I'd like to thank the staff, Dan and Ty, for all the work that they've put into this project. We've been very lucky to have a lot of free accommodation on the island. Thank you to the Short Smiths and the Paytons, and also to Sea Link, who've provided us with some free ferry rides. And there's also been donors to the Bushfire Appeal and the orchid work, and especially the French company Carabos, who donated profits from their Fire Gardens show in the Adelaide Gardens. We have a lot of volunteers that have been involved. So for field work, thanks to Denzel Murfitt, who travels with us on a lot of field trips and has amazing expertise. And also on the island, Alison Buck, Andy Young and Michelle Haby have been some of the botanists that I've talked about that have really helped us locate a lot of these populations. In the laboratory, there are a number of volunteers, Thelma, Tiffany and Laura, who are helping with the propagation, and also Cale Gordon and Jake Thornhill, who've been helping potting out into the shade house. Okay, that's the end of my talk. Thank you.